So we left off, I believe, at the satellite cell, the last cell that we were going to cover when we were talking about neuroglia. Last lecture, just to review briefly, we introduced neurons and neuroglia. Neurons can be categorized based on their function. We have sensory, we have interneurons, and we have motor neurons. We also can categorize neurons based on their structures, particularly how many branches they have coming off of them. We can have the unipolar, the dipolar, the multipolar. We talked about branches of the nervous system. We have the central nervous system, which is the brain and spinal cord. We have the peripheral nervous system, or PNS, which is going to be everything outside of the brain and spinal cord. And within the PNS, there are the subdivisions of the afferent and efferent subdivisions. The efferent subdivision can be broken down into autonomic and somatic. And then the autonomic subdivision of the efferent division can be broken down into sympathetic and parasympathetic. And we'll talk about those subdivisions in more detail as we progress. That was just kind of a big overview. As we think of neuroglia are the support cells of the nervous system. They help the neurons to do their job. We talked about 4 neuroglia that are in the central nervous system: the oligodendrocytes, the astrocytes, the ependymal cells, and the microglial cells. And then we talked about 2 neuroglia that are present in the peripheral nervous system: the Schwann cells and the satellite cells. 2 of these neuroglia are going to get a little bit more attention: the oligodendrocytes and the Schwann cells. We're going to give those a little bit more attention because they are responsible for making myelin, for myelinating the axons or nerve fibers within our bodies. So when we think of myelin, guys, gals, I want you to think of it as an electrical insulation. And it's going to make it so that we will have our axons transmit information quicker. Really pragmatically speaking, the more myelin that's on an axon, the faster the action potential conduction speed will be. So you can send that signal faster with myelin. Without myelin, that signal is sent slower. And when we look at myelinization, just to reiterate, in the central nervous system, the oligodendrocyte is the myelinization. And the peripheral nervous system, the Schwann cells are responsible for myelinization. And as we're looking at this myelinization process, we aren't born with fully myelinated axons. This process takes time. You may notice how, and this is just classic child development, when kids are born, they have terrible control of their bodies as an infant. And then they become a toddler and they walk around and they're jerky and they're cute. And as they age, they become more coordinated. This increase in coordination corresponds to an increase in myelinization. They can send the signals quick enough to make the adjustments they need so they don't tip over while they're trying to walk. One of the reasons why it's important to get enough calories in our diet is because we use particularly dietary fat for myelinization. So when we think of breast milk, there's those campaigns, breast is best. There's a lot of dietary fat present in breast milk, particularly in colostrum, the first milk that's secreted. When you get to that stage of life and you have little kids that are transitioning from milk to solid food, pediatric guidelines typically stipulate that you give your children whole milk and not skim milk because they still need lots of dietary fat, particularly milk fat, to help with this myelinization process. Now, as we look at the Schwann cell, I want you to think of the Schwann cell kind of like a cinnamon roll, in that one individual cell will wrap itself multiple times around an individual axon. And as it's wrapping itself multiple times around an individual axon, most of the fat of this myelin is going to come from the cell membranes of this Schwann cell. The repeated layering of the cell membrane is going to contribute a lot of lipids to this myelinization process. But the outermost layer, or the outermost wrapper of this myelinization process of the Schwann cell has a special name. It's called the neurolemma, or neurolemma, I should say. And we call it the neurolemma because it's going to have the nucleus, it's going to have the organelles of the Schwann cell. This is the part of the Schwann cell that really is the nuts and bolts that keeps the Schwann cell alive. And then outside of this neurolemma, we have another layer, the endoneurinium, which you should have been able to identify in lab up until this week, unless you have the Friday lab section. We'll talk about it today during lab. Within the central nervous system, we have oligodendrocytes. One oligodendrocyte can myelinate multiple portions of multiple axons, multiple segments on multiple axons, we'll say. So when we look at the oligodendrocyte, instead of wrapping its entire self around the axon, it's going to have a pseudopod or projection coming off of it, and that pseudopod-like projection that comes off of it will wrap itself around an individual part of an axon. A key difference between an oligodendrocyte and the Schwann cell is the CNS versus PNS distinction. You need to make sure you commit that to memory, because a lot of test questions are going to be dependent upon what or where those individual glial cells are going to be located. Are they CNS or PNS neuroglia? Now, when we look at nerve fibers within the central nervous system, sometimes they're called tracts within the central nervous system, nerves in the peripheral tract in the central nervous system. We don't have the neurolemma because we aren't going to have all of the cytoplasm, the nucleus and the organelles on that outermost layer. With our oligodendrocytes, the nuts and bolts of the cell are in a separate cell body, not wrapped around the outermost layer, like it is with the Schwann cell. So, no neurolemma. Another key difference is that in the central nervous system, we're not going to have the endoneurinium. Endoneurinium is only going to be present in peripheral nervous system nerves. It will not be present in central nervous system tracts. Now, as we're looking at myelin, most of the myelinization that we're going to study is going to be within the peripheral nervous system, with Schwann cells being wrapped around an axon. And there's a little gap in between the Schwann cells. This little gap is known as a node of Ranvier, or Ranvier. It's a French word. I'm not sure if I'm pronouncing it correctly. Your lab manual, this edition of your lab manual, introduced a brand new name for it I've never seen before, but it's a good name. Myelin sheath gap. It's a very descriptive name for this space in between the myelinization of an axon. Most figures that you look at in textbooks will have it be very overemphasized in terms of its diameter or the width of this gap. But you may have noticed when you're looking at your longitudinal nerve sections during lab, this node of Ranvier is itty bitty. It's super tiny relative to the width of the Schwann cells themselves. Now, as we're looking at the node of Ranvier, this is the part of the axon that gets electrified. And if we have this myelinization, the signal will travel faster. Now, one of the things we need to emphasize with these action potentials that we're building up to is that action potentials on our axons all originate at the same spot. They are all going to originate right here at the beginning of the axon. This area of the axon is the axon hillock. I didn't mean to cross that out. I meant to underline. Axon hillock is where those action potentials originate. It's known sometimes as the trigger zone. And then from there, that action potential will proceed unidirectionally, or one direction, down the axon. We also have some nerve fibers that are unmyelinated. And I remember being particularly confused about these unmyelinated nerve fibers when I was a student, because they still have Schwann cells associated with them. So as we look at this unmyelinated nerve fiber, the thing that makes it, quote unquote, unmyelinated isn't necessarily the presence or absence of myelin, but it's those nodes of Ranvier. As we look at a myelinated axon, it has there are little segments that are completely surrounded by myelin, and the action potential jumps from internode to internode, or node of Ranvier to node of Ranvier. Now, with the unmyelinated axons, we're going to have a strip down the length of the nerve fiber that's completely unmyelinated. So here's a segment right here that's unmyelinated. Here's a corner right there that's unmyelinated. And because we have these strips that are completely unmyelinated, instead of the action potential being able to jump from internode to internode, the action potential has to move down the entire length of the axon. So I really want to emphasize unmyelinated nerve fibers don't have nodes of RON-VA, but they still are myelinated by variation of the Schwann cell. So let's have a review question. Which glial cell is found wrapped around nerve fibers in the peripheral nervous system? The oligodendrocytes, the Schwann cells, the satellite cells, nodes of RON-VA, or the ependymal cells? So we're down to about 20 seconds. Make sure to get your answers in. The correct answer is Schwann cells, and as a class, we did very respectable. Nicely done. So let's talk about how we conduct the speed, or how we can move those action potentials. There's two methods. We have the fast method and the slow method. If we have the action potential moving quickly, that's typically going to be in a large diameter axon that has a lot of myelin. So the wider the axon, the faster the signal. The more myelin, the faster the signal. So as we look at being myelinated, that's going to speed up our signal. And as we look at the diameter, that can also influence our signal speed as well. So let's talk about some numbers. Our small unmyelinated axons can send the signal at about 0.5 to 2 meters per second. So not terribly fast. Our small myelinated fibers can go up to 15 meters per second, and then our large myelinated fibers can go up to 120 meters per second. Now, in terms of where we have fast signal speeds and slow signal speeds, we want to have fast signal speeds primarily in two parts of our body. We need those fast signal speeds associated with sensory perceptions that will help us to avoid bodily harm. So we have lots of fast signal conduction with pain receptors. We're also going to have lots of fast signal speeds for skeletal muscles. We need to be able to coordinate and control our skeletal muscle nearly instantaneously, because if we can't coordinate and have that quick signal speed to our skeletal muscles, we'll be very uncoordinated, very clumsy, and have more damage to our body incur. So to go back to the toddlers that are in the process of being myelinated, one of the reasons why kids that are just learning how to walk are so clumsy is because they don't have the myelinization yet of their large motor neurons. So they have poor coordination. They can't send the signal as quickly as they need to in order to be coordinated. When we think of where we want slow signals, as a rule of thumb, we want the slow signal conduction speed to be associated with something that's not going to be immediately life-threatening. So those are typically going to be signals from the visceral organs, or the organs of the thoracic and the abdominal pelvic cavities. When we look at a nerve fiber, or an axon of a neuron, we can regenerate these axons. And within the peripheral nervous system, this is old news. Regenerating axons in the peripheral nervous system has been widely established. It happens all the time. You may know somebody who's had a surgical incision, and immediately around that surgical incision, they have an area that's numb. They can't feel anything. But as the days, weeks, months, and years go by, they regain their sense of perception. They can regain their sense of touch around that surgical incision. This is nerve regeneration at its finest. In 2000, 14, so still fairly recently as far as I'm concerned, there were several growth factors that were isolated to stimulate axon regeneration within the central nervous system. And this is exciting because if you think of spinal cord injury, repair and recovery, that's kind of the holy grail right now of nerve regeneration. We're not quite to that point. But let's talk about how it works in the peripheral nervous system. So step number one is that you're going to have some kind of damage occur. You cut the axon. And this is going to cause downstream death or destruction of the axon. So we're going to find, after that cut is made of the axon, the downstream or distal portion of the axon is no longer going to have transport vesicles delivering nutrients. There's no longer going to be fast or slow axonal transport. And that downstream portion needs to be recycled. So we'll have macrophages move in, clean it up, and move it out, and remove the debris. However, the Schwann cells are still alive. And those Schwann cells have that hollow tube that's left over after they've been wrapped around the axon. And as the axon is regenerating, the Schwann cells themselves guide the regeneration of the axon. They help to direct its growth back to where it was supposed to be. Now, to speed up this process of regenerating the axon, we find that the neurosoma, or the cell body of the neuron, is going to swell up. And this swelling of the neurosoma is going to increase the intercellular pressure to help push the axon farther away, to help accelerate the growth of the axon. One of the things that we're going to need is nerve growth factors. These nerve growth factors, we'll talk about it in a little bit in a couple slides, they help to direct the growth process as we're regenerating these axons. So if we don't have any nerve growth factors present, the neurosoma is not going to can be nice and compact. The nerve growth factors prevent it from swelling up. So this neurosoma swells up. That causes the axon to get pushed farther away. As the axon's getting pushed farther away, it's going to move through the tube made by the Schwann cells. And as it's moving through that tube, it'll eventually reconnect to its target organ. And once it gets reconnected to that target organ, it's re-exposed to the correct growth factors. The neurosoma shrinks back inside. It no longer needs to be swollen up to push the growth of the axon. And as we're going through this process, you know, in the peripheral nervous system, yeah, there's an okay success rate. When you think of how unbelievably complicated this is and how many wires or axons are regrowing for the electrical circuits of our body, the fact that this works at all is pretty impressive to me. However, it's not a perfect process. When you have large incisions, large wounds, not all of the axons will reconnect to the correct target organ. A lot of them do, but there's not a 100% success rate. So there's some things that can influence the success rate. If you have a high density of axons, or another way of thinking of it is, if you have a lot of regeneration tubes that the axons can grow through, sometimes the axons grow through the wrong tube and then will reconnect to the wrong organ. But if you're working in a part of your body that has a low density of axons, there aren't a lot of options for where that axon can regenerate to, and you're more likely to have success. So practically speaking, if you have an incision, maybe in your skin or your skeletal muscle tissue, that incision, those axons have a high chance of regenerating and reconnecting to where they're supposed to be. But when you have damage to, say, the spinal cord, where there's an unbelievably high density of axons, getting that one specific axon to reconnect to that specific part that it needs to go to when there are billions of options all clustered right next to each other is statistically really, really difficult to do, which is why we have such a hard time recovering from spinal cord injuries. This nerve growth factor, NGF, is one of the chemicals that are primarily responsible for directing the growth of axons. And nerve growth factor is kind of an umbrella category. We're finding more and more variations of nerve growth factor as more and more research has been done. When we look at nerve growth factor, it primarily is going to take that neuron that was damaged and allow for its axon to reconnect to the target organ. So it's going to help connect the growing neuron to the target organ. So let's have another review question. In order to regenerate, a peripheral nerve fiber must have which of the following? All of the dendrites intact, at least half the length of the fiber intact, at least the entire axon and neurolemma intact, the neurosoma and some of the neurolemma, at least part of the neurosoma. So when we think neurosoma, think cell body of the neuron. When you think neurolemma, think of the Schwann cells. We're down to about 10 seconds. Make sure to get your answers in. And remember, this is just participation. It's okay to answer these wrong. For a nerve fiber to regenerate, we need the cell body of the neuron and we need the Schwann cells intact. So when we think of cell body of neuron, I want you to think neurosoma, and that neurolemma is associated with the Schwann cells. So to back up a slide here, we need to have the cell body, and then we need the neurolemma, or the Schwann cells, to guide the growth and direction of the axon as it's regenerating. And that is all for this presentation. Let's go ahead and flip to the next one.